think we've gone through most of the vocabulary at this point. Today we're going to focus a little bit towards the end, or maybe to next class, on AFLPs and RFLPs, the bottom two there on the left side. And we may or may not get to non-disjunction this topic. Not just this class, but at all. We'll see. Does anybody have any questions before we get going? Anything I can clarify for anybody? Concerns? All right. So last time we got together, I went through one example. I just want to briefly revisit. The concept here is that PCR starts with one or a few, maybe, double helices. So initially, these two strands were part of the same molecule. If you want, double check me on your device or later that these two DNA single-stranded sequences are antiparallel and would base pair with each other. The representation of one molecule that's been denatured, heated to about 95 degrees centigrade to melt the strands to make them single-stranded. What happens next? We're going to make more copies of this artificially using the PCR technique. What's the next thing we have to do? DNA synthesis starts with?
Priming.
Yeah, we have to add primers. Shown here, that are designed to be complementary or base pair with the template DNA. And the 3' ends, then DNA polymerase will do the third step, which is on the next slide, which is extension. It will copy, using the top strand and the bottom strand, the full initial chromosome sequences, the ones shown in gray, as templates. It will add the complementary nucleotides to synthesize the strands. So we've gone in one cycle from one double helix to two double helices. And that's what's shown here, which may be a little confusing. And I went over a little bit, maybe too quickly last class, maybe. So quick review of this. Two important points to note. One is these are two molecules of DNA from the same person. And when that second strand here on the top, when DNA polymerase grabs onto the end of that primer and finishes completion, that's this step of extension. Start with that primer sequence, and this diagram beneath it is what that primer would look like after it has completed synthesis. DNA polymerase has added all the black nucleotides. So at the end of the second round of PCR, then not only do you have those two templates, you've got two new templates on the bottom, the original one and the newly synthesized one in black. So you've gone from one set of double helices, or one set of strands, to two. You've got twice as many primer binding sites for the next round of DNA synthesis, which is what's shown by these new primers, which can base pair with that newly synthesized DNA. So in other words, we've gone from one copy of the primer site down here, TAA, G, so forth, to a second copy up here, same sequence. It was just synthesized. Now in the next round of synthesis, there are places for two of these primers to base pair and make more strands in the following cycle. What does this mean about primers and PCR? How many primers do we have to do PCR? We have to have two different sequences of primers, but in terms of, dare I say it, moles of primers, dun dun dun, chemistry. In terms of actual number of primers, we have two different sequences, but how many of each of those do we have to have in the test tube with the DNA to do this process? Lots. Right. Generally speaking, it's not two, as shown on this screen. This is just the first round. Would use two pieces, two individual molecules of primers, but after you do this 20 times and you do exponential growth, you do literally need millions plus of each of these two primer sequences, because every time you start synthesis of the second strand with a primer, you have to have a molecule of that primer. you don't need to know this until you get to Biology 104 or until you start working in somebody's research lab, but just to give you an idea of how this actually works in the laboratory, we usually set up 10 microliter PCR reactions, with a microliter being a millionth of a liter. So tiny little drop of liquid, 10 microliters, that contains what's written up here. So literally, 6 trillion of each of those two primers. In that volume, 10 microliters, 6 trillion of each primer plus about 47,000 copies of each chromosome. And so in that 10 microliter, in that tiny little volume, there's a huge concentration of these primers. Trillions of them. And that's how we can use exponential growth. We have to have that many primers to go from 2, 4, 8, 16, 32 by 25 or 30 cycles. You're up into the billions of copies of whatever piece of DNA that you're trying to make copies of. Nothing on that is going to be on the test. That's just for perspective. So if you're going to set up a PCR reaction, last bit about PCR, then we're going to talk a little bit more about analysis. We need these 3 parts that are DNA. The template, double helix, a forward primer and a reverse primer, trillions of each. What else do we need to add to get DNA synthesis to work? We've already talked about one of the 2 other things last class.
Polymerase?
We need an enzyme, DNA polymerase. So we buy it from an enzyme supplier. Actually, I buy it from New England Biolabs, where we also learn about our restriction enzyme sequences. So DNA polymerase, you can buy it from a manufacturer. They isolate it and purify it and then sell it back to us at exorbitant prices for tiny little tubes of it. The other 2 things we haven't talked about before, but are really critical for DNA, well, one is critical for DNA synthesis, the other not so much. So I'll give you number 5, it's a buffer. So if you're actually in the lab setting up a PCR reaction, you would put template DNA, primer 1, primer 2, tack, buffer. Buffer is just to keep the salt concentration right and the pH right for the polymerase to work in. It's very finicky, enzymes are. You need a certain number of salts, certain types of salts, concentration of salts. There's one more critical thing that we've never talked about before. What do you need to create new DNA? After DNA polymerase binds the 3' end of a primer, do you build DNA from scratch? It's just invented out of thin air.
Nucleotides and sugars?
Nucleotides, yeah. So we need to add the building blocks, the A's, T's, G's, and C's. And you can imagine that's probably billions, trillions, quadrillions of each of those too. So we're synthesizing so many millions of copies of this sequence, we need loads and loads and loads of that individual nucleotides. That's the last thing. All critical. If you don't add any one of these things to our reaction, it won't work. So agarose gel electrophoresis, hopefully you've seen the video. Does anybody have any questions about gel electrophoresis? We're going to go through a couple of examples of how it works and analyzing PCR products digitally on slides. Any concerns? Okay. So what is it and why do we use it? What's the goal of agarose gel electrophoresis? Better be careful when you're scratching your face because I might think you're raising your hand. Go for it.
It's to separate the different sizes of DNA lengths to try to identify what it is.
Okay, so separates DNA by length. Okay, somebody in the back, 1, 2, 3, 4, 5 rows. That's you folks. I don't hear from you a lot, so here's an opportunity. What's a gel? What do I mean by gel electrophoresis? Name something that's a gel. Hair product. Any other thoughts? Pardon? Right, so an agar, so when do we use agar? Not agarose, this is agarose, but when do we see agar? That's also a gel. In the lab, like Petri dishes. That's a gel. Agar media. You poke it and it's kind of spongy, but not quite. What's a common gel to eat? Jello, made out of gelatin. Agarose gel electrophoresis is basically Jello or agar. It's a matrix. When it's hot, it's liquidy. You melt it in a microwave or you melt it in an autoclave, pressurized microwave. Well, not really. And when it cools, the molecules of agarose or agar form a matrix. They solidify, they form a regular structure, kind of like Jello, it's kind of wobbly. And what we do then is we add DNA molecules, double helices of different sizes. So we have a long molecule and a short molecule. So many base pairs on the right, few base pairs on the left. And we get this DNA into the gel somehow. We shove it into the gel. And then the DNA has to move through this gel. And it does so according to a particular principle, which is what I've asked a couple of times now on Socratic exercises. How does agarose gel electrophoresis separate molecules of DNA based on size? Charge, polarity, anything else? There are two things that are critical. There were kind of two right answers. One of them is charge. Oh, go ahead.
The gel, kind of danger Jello. It's thick enough that you put it in for a certain amount of time and only certain things will be able to get through it faster. So the small pieces can go faster and the long pieces stay near the end.
Okay, so here's an important point. Combining these two thoughts. One is charge. What charge does DNA have?
Negative.
Phosphate backbone is negative. So there are negative charges on both of these molecules. So the electrophoresis part, phoresis means move, electro means we're going to zap it with electricity. Which direction is going to DNA? Which direction is DNA going to move? Is it going to move towards a negative pole or a positive pole?
Towards the positive.
Opposites attract. So DNA, what we're going to do is we're going to get this Jello, we're going to put it in a buffer tank that's covered with liquid that is conductive of electricity. We're going to attach a voltage supply so that the positive terminal is on the far side of the gel from where the DNA is. So the DNA is going to be attracted to the positive pole. It's going to move this way into the gel towards the positive pole. And then the point made down here is, and this is always confusing a little bit sometimes, which molecule gets farther faster? It's the small one. So does anybody disagree? Because I would be happy to explain why it's not the opposite. There's a common alternative conception here about which one will move first. But the theory is, or the principle is, that the holes in the agar, in this mesh, keep the big molecule from moving faster through it because it's being impeded by all of these molecules of agarose. While the small one can fit in between the holes in the grid of agar more easily, so it moves farther faster. It doesn't have critical point. It's entirely due with the length, the number of double helices, that is the number of base pairs. It's the physical size. It doesn't have anything to do with the composition of the nucleotides. It doesn't matter if it's A's, T's, G's, or C's, because the charge is on the backbone of the molecule, not in where it base pairs, not the nucleotides. It's the phosphate backbone that matters. So smaller moves farther through the gel than large fragments. So that large double helix would not move very far in a fixed period of time, while the small one would go farther through the gel. And depending on how thick you make the gel, that is the percent of agarose, you can differentiate between very, very similar sizes of DNA, in the order of maybe 10 to 50 base pairs. So it's pretty precise in terms of the sizes of DNA that you can differentiate. And that's important because we're going to be talking about STRs and microsatellites and distinguishing chromosomes that differ with very little length. And here we are. So back to this. Take a minute, please, maybe 3, depends on how long it takes, because it's just counting nucleotides. We need to figure out, remember, this individual is a heterozygote. They had an allele 6 at TH1 and an allele 7 at TH1. So if we zoom in, we see that there are some copies of allele 6. One of the alleles had the 6, that was the top strand, and the bottom strand was a 7, which means that those should be 7 and 6 and 6. So all the copies of DNA, in other words, that are made from this chromosome that has 6 repeats of AATG will be different in size from all of the PCR products that are made from the bottom strand, which had 7 repeats. The question is, how long are these 2 molecules of DNA going to be after PCR? When we amplify using these 2 primers, how big is that piece of DNA that's created from both chromosomes? So take a couple minutes. It's just counting nucleotides. Remember to include the number of repeats. It's either 6 or 7, and that's what's going to make all the difference. We just need to know. We're going to load a gel next digitally on the slides. We need to know what we expect the lengths of these 2 PCR products are going to be. Does everybody understand the instructions or have questions about this? Okay, go for it. How many nucleotides are in the repeat in this allele?
24.
There's a repeat of 4 nucleotides repeated 6 times, so that's 24 nucleotides. Each of the 2 primers is 24 nucleotides. So that amplified piece of DNA will be how long? It's going to be the primer plus whatever was copied in the middle between the 2 primers plus the length of the other primer, which is 24, 24, 24, 72 base pairs. So do the same thing for the bottom. How long is that fragment going to be? +, so it's got an extra copy, so it's 4 times 7 instead of 4 times 6, so 24 plus 28 plus 24. The 2 only differ by 1 repeat of 4. 6 copies here, 7 copies there, so this one will be how many longer? 4 longer, because there's 1 extra AATG that gets copied as part of PCR. So instead of 72, 76 nucleotides. So we start with 1 suspect or sample of DNA. If they're heterozygous for an STR or an indel, some sort of polymorphism that changes the size of the chromosomes, if we have primers that flank that difference in size, we're going to make 2 different sized pieces of DNA during PCR. So now that the PCR product is created from a heterozygote, we know that agarose gel electrophoresis separates by length and charge. Charge is essential. Everything that's DNA is negatively charged. If it wasn't charged, we wouldn't be able to do gel electrophoresis. But the primary thing that separates based on size is not charge, it's the length. So charge matters, but that's not what this causes the separation of sizes of DNA. It's the length of the molecules that matters. So here we go. This is an agarose gel, at least a diagram of one. You've seen this in drawings in the movies and so forth. So at the top we have diagrams of 2 depressions or indentations in the gel. So think of this as a three-dimensional slab that has a little hole in one end. There's one hole there that we're going to allow DNA ladder, or M for marker, which is a typical abbreviation, marker. It's a composition of different DNA band sizes that, again, we purchase from a supplier, and we know that the bands, that the DNA products that are in that tube have specific sizes. So they let us, they're a standard. They let us know how big the molecules we produce are by comparison to this banding pattern. So in this case, it's a 10 base pair ladder. There's a band every 10 base pairs with the exception of 30 and 50. So the idea here is load that 10 microliter PCR sample into the empty well, apply a voltage. What goes at the bottom of the gel in this example? Negative or positive at the bottom? Are you positive? Yeah. Right. DNA is negatively charged. It's going to migrate. You want it to migrate through the gel towards the positive terminal. So negative at the top, positive at the bottom. DNA goes in the well, 10 microliters of sample, with a colored dye. So we add basically a navy blue dye that's also a charged molecule, so we can see where the DNA is as it moves through the gel. It doesn't stain the DNA, it just shows how migration is progressing through the gel once the electric field is applied. And where should our two molecules wind up? We just predicted what the band sizes or what the DNA PCR products were going to be from this PCR. It was 72 and 76. So we'd expect one band that's close to 70, that would be the 72, and something that's right there. This is probably something I'll ask you to do on some sort of an analysis, like an exam. Predict. Here's a DNA molecule, here are where the primers are. What are the size or sizes, depending on whether or not it's a homozygous individual or a heterozygous individual? What are the sizes going to be, and draw a gel that predicts what you would observe if that's true. So here we get to one of our first vocabulary terms for today, amplified fragment length polymorphism, or AFLP for short, acronym. That's one of our vocabulary items. Amplified, let's break it down quickly. Amplified, that means it was produced by PCR, amplification. We made lots of copies of something. It's a fragment, so that's a PCR product. Fragment meaning short piece of DNA. I'm not going to define length for you. What is polymorphism? What does poly mean? Many. And morph means form, like many forms. A polymorphism is a general term that geneticists and evolutionary biologists use to indicate something that differs between molecules or between individuals. A heterozygote, in other words, is polymorphic. They have two, more than one, not many, but two. A heterozygote has two alleles. So that's a polymorphism. There are two different versions of the molecule. And in this case, we see that when we run this agarose gel, and we observe two different bands. Does anybody have any questions on this process? We've looked at where primers base pair with the DNA template, try to figure out what size, calculate the number of nucleotides are in the two primer sequences plus the number of nucleotides that are in between them. That gives you the total length of the amplicon, the fragment that gets generated, and then we electrophoresis them and see where they are relative to DNA ladder. Quick check. Blue band, how many base pairs?
30.
It was actually 31, but I'll give you 30. Play with me, come on. Okay, so good. Now it's your turn again. Genotypes, PCR, gel electrophoresis. I've listed 3 different genotypes up here, TH6, TH6 over TH7, and TH7 over TH7. This is what kind of a genotype? Homozygous, heterozygous, homozygous. We just did the PCR for TH16 over TH17 after PCR. So these are all the same PCR, same sequence, same STR. We just drew this one in the middle. Please draw it again. Label the bands, so drawing the 2 bands wherever they were supposed to go. Label what sizes, in terms of number of base pairs, they were. And when you're done with that, draw what a homozygous individual of both the homozygous genotypes would look like after electrophoresis. So the middle we just saw. How do we expect this to differ between a heterozygote and a homozygote? If you're homozygous, then
And we would only have one band on both of the homozygotes at 76 and 72. 76, 72.
yeah. So this TH1, which allele is bigger, TH16 in terms of number of base pairs, 6 or 7?
7.
7's longer, so it's 70. That's a 76 nucleotide allele and a 72 nucleotide allele. If you're homozygous, then you've got the maternal and the paternal versions of the chromosomes, but they have the same allele. So how many bands will there be? One. Fantastic point. So you've got twice as many. If you're a heterozygote, you have, say, you've got 1 copy of the TH. Let's say you got a single cell from a suspect. Has 1 double helix from mom and 1 double helix from dad. So there's 1 double helix that's TH16 and 1 that's TH17, and you get bands that are this thick. You get an equal amount of product from both of the 2 chromosomes. They're just different in size, in physical size. So if you start with, drawing that's 1 and 1. So if you have the same for TH16 homozygote, you've got 2 copies of the same size chromosome. So you do only get a band that's the 72 base pair band. But there's twice as much DNA of that size, because here, the 6 was generated from a single double helix, and that larger band, the TH17 allele, was also generated from a single 1 double-stranded DNA molecule. Over here, you've got 2 template strands that are the same, so you produce twice as much of the same size PCR product. Unfortunately, when we do PCR in real life, this sort of difference isn't as obvious, and I'll show you an example of a gel. You can see it if you want to skip ahead to the next slide. The band intensities aren't always as obvious, but indeed. The premise is true that if you have twice as much template, then you should produce twice as much PCR product. So what does the band on the right side look like? TH17 homozygote, how many bands? Just 1. There's only 1 size of amplicon, 76 base pairs. So it should be the same size as, those should be the same size as the heterozygote band sizes. Same chromosomes, just in different individuals. Homozygotes, heterozygotes. This is how the DNA evidence and DNA technician is analyzed using these CODIS STRs, the ones that scientists, forensic biologists use to analyze DNA samples. If someone's a heterozygote, you can see different band sizes on the gel. If they're homozygous, 1 band. Heterozygous, 2. So in this gel, the AF16 chromosome between the 5' ends of the primers is 700 base pairs. Between the same primers, when you look at a different template, the same chromosome but in a different strain, is 1,000 base pairs, and that's why we see differences in sizes on the gel. And that's an AFLP. The last piece of vocabulary that we need to do is a restriction fragment length polymorphism, which is an RFLP, which still means there's going to be a polymorphism. There's still going to be a difference in size, but it's not based on PCR alone. We have to do something else to this. This was the activity I asked you to work on for today. 2 different versions of a chromosome, 2 alleles, 2 homologs, paternal and maternal. Find a heterozygous nucleotide, design a primer pair, predict how long that amplicon will be, how much DNA is going to be synthesized using the primers you designed. And then lastly, something about a restriction enzyme. So, where's the heterozygous nucleotide? Let's go to the next slide. So where's there a SNP, polymorphism between the paternal and the maternal homologs?
The paternal has 3 C's.
Paternal has 3 C's, the maternal has 2. So there's a difference right there, and then of course on the corresponding second strand. There's been a CG mutation. One of them is a C, the other one's a G. So there's a C there, but a G there. What sort of primers? Did anybody actually come up with a primer sequence that they want to use? Feel free to share. You're among friends. What part of these chromosomes would you design one of the primers to? The right side of the top strand, the left side of the top strand, left side of the bottom strand, right side of the bottom strand. Yeah.
It would be the left side, or the right side of the top and the left side of the bottom.
Okay, so I heard right side of the top, left side of the bottom. That's given an assumption. That's how we drew it last time. There's one assumption that's made.
It's 5'.
Right, so assuming that the top strand, as is typical for genetics, is in the polarity 5' to 3'. First, we have to know the polarities of all the strands before we design primers, because we need to make sure that the 3' end of the primer is pointing in the direction we want DNA polymerase to synthesize the DNA. So, right side of the top strand, 5' means that our 3' end is going to be pointing that direction to synthesize across the region that we're trying to make copies of, so that's perfect. Likewise, on the bottom strand, the primer would start someplace on the left and go then to the right. So how do you make a primer? How do you design a primer now that we know where they're supposed to go? What's the other property of a primer we need to know?
You need to know what you're actually trying to amplify.
Okay, so in this question I said, let's amplify this entire piece of DNA, which is 3, 6, 9, 12, 18 nucleotides long. Remember, what property of the primers defines how much DNA you're going to copy? Exactly the outer edges of the amplicon are defined by the locations of some part of the primer. The 5' ends. So if we want to copy this whole thing, the 5' end of this primer has to start at the first nucleotide we're trying to copy. So what's the primer sequence going to be? We need this primer to base pair with the top strand. So that's close. It's right. So it's going to be the same sequence as the bottom strand because both the bottom strand and this primer have to complement and base pair with the top strand. So the primer would be AAA, T, T, T. It doesn't have to be 6 nucleotides. You can make it however many you want. You could make it 3, 2, 7, whatever. That wasn't specified in the question. So that'll put the 3' end of that primer there. Polymerase can synthesize the rest of the second strand based on the top strand template. And again, because this is the way I wrote this question, the other primer sequence happens to be the same. You can make it 4 nucleotides if you want to. AAAT, 3' end. And that'll copy across using the bottom strand there as the template. And you can use the same primers on the bottom 2 strands as well, down here, the maternal copies. If we do this, then, if we add these primers and we use them to do PCR on this sample, is this a heterozygous or a homozygous individual? Hetero because there's a SNP, right, that single nucleotide polymorphism. There's a C on the paternal copy of the chromosome. There's a G at the same spot on the maternal homolog. So this is a heterozygote. How long are these PCR products going to be from this one individual sample? Are they going to be different lengths or the same length?
18, 17.
Let's see. So the 5' ends define the amount of DNA that's going to be copied. So they should both be the same length. Because there's no STR, there's no microsatellite, there's no difference in size between the 2 homologs, there's just a difference in one nucleotide. It's there, it's just a G on one homolog and a C on the other homolog. So no size difference this time. Both of these are going to be 18 nucleotides long. So what happens then? What did that C to G mutation do? It didn't change the length of the chromosomes, but that C to G changes in a very specific spot that changes something from one homolog to the other.
Changes the binding sequence?
It changes some sort of sequence. So what is that sequence? CCC-GGG is what sort of a sequence? It's a palindrome. So that's a restriction enzyme binding site. On the top strand, there's a palindrome, or sorry, in the paternal homolog, there's a palindrome. CCC-GGG reads the same on the 5' to 3' on the second strand. Is that binding site there in the maternal homolog? CCC-GGG. Because of this point mutation, there's no binding site for a restriction enzyme there. And that's the restriction enzyme that's asked about. The last part of this question is what happens to these 2 pieces of DNA if you add the enzyme small1? So hopefully by now you'd be comfortable looking up small1 and finding that it does bind CCC-GGG and cuts in that sequence. Yeah.
It would cut the paternal sequence in half.
Right?
So the CCC cut GGG.
That's correct.
But the maternal one, it wouldn't do that.
So it will cut, small one cuts right in the middle of the CCC-GGG from both strands. So how long would those, after digest, how long would those pieces of DNA be? PCR amplify 18 base pairs. And then you add this enzyme. It's going to cut the paternal PCR products into what size pieces?
9 base pairs.
9 and 9. But does it cut the maternal amplicons, the pieces of PCR product that are produced from these templates? No binding site for small one, the enzyme, so no cut. So that will produce, after digest, the whole 18 base pair fragment is not cut. So for next class, right. Try adding those to the next two wells in the slides. See what would the restriction digest and then PCR agarose gel electrophoresis patterns look like. Here's the key. Minus, that is without the enzyme on the left side, what would it look like without the enzyme? That is just the PCR with no digest. And then on the right side, what would happen, what would it look like when you add the enzyme and then run the gel? Without small one on the left, with small one, the digest enzyme. And we're going to get into the next topic next class. So aside from that, please take a look at the next chapter. And we'll start with exercise 8.1 as well next class.